Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. This video is a continuation of a video that I began yesterday on cellular transport, particularly on active transport, but my camera shut off and I didn't have my charger with me. So please forgive me, but I'm going to pick up where I left off. In the previous lectures, we were discussing passive transport, osmosis diffusion, and facilitated diffusion. You should understand those concepts. We're going to test heavily on that stuff. You need to know about tonicity, hypertonic, isotonic, and hypotonic, and what they mean, what would happen to red blood cells if placed in those solutions. And then we're going to we began discussing ATP, a high energy compound that our cells use to digest, to release energy, and that energy can be used to perform other functions. Now, I began talking about one form of active transport called the sodium-potassium pump, and that's where I'm going to pick up here, okay? So, in passive transport, if I had a membrane separating two areas, and we drew this and we talked about it. If I have a membrane that is semi-permeable, and I have a low concentration of a substance on one side and a high concentration on the other of the same substance, if this membrane is permeable to that substance wants to move across the membrane until it reaches an equilibrium. That passive movement is diffusion. And we can always draw the concentration gradient from the high side to the low side and predict which direction it would move. If the substance cannot move, osmosis usually does in the opposite direction. But what happens in our body if I have a high concentration here and a low concentration here, but I need to move some of this stuff back across against the concentration gradient? It's just like swimming upstream or paddling upstream in a river. You're going to have to burn energy to make that happen. One of the ways we do that is the sodium-potassium ion exchange pump, or the sodium-potassium pump. It's an important concept because later in the semester we're going to talk about other ion pumps, especially calcium pumps, and how they help our body maintain homeostasis. So now, if I had a small cell here, and I cut a section of the cell membrane out and I magnified it so that we could see it really, really well, what we'd see is that lipid bilayer, that fluid mosaic model that we discussed in the previous lectures. Embedded in the lipid bilayer would be several integral membrane proteins. So if I were to look here, I'm leaving a gap for a reason. I have a protein that's embedded in the cell membrane. I'm actually going to draw three different ones here. On this side, I'm going to add a little ion channel. There are proteins that span the membrane and form little channels that can open and close and allow ions to pass through or allow substances to diffuse across the membrane. Sometimes those channels are completely open all the time and allow for passive diffusion to occur. And back in the 1940s, some scientists measured the concentrations of certain ions in our body fluids, in the extracellular fluid and the intracellular fluid. One of the things that they realized, or that they measured, was that the concentration of sodium is much higher outside the cell than it was inside the cell. So if this is outside and this is inside the cell, then the outside concentration of sodium is higher than the concentration of sodium inside the cell. Anytime you see these little squared off brackets, that means the concentration of whatever's inside of there. So, given that this ion channel will leak sodium and I have an imbalance from outside to inside, I can see that the concentration of sodium is much higher outside than in. Sodium ions would begin to diffuse into the cell. And as they diffuse into the cell, those sodium ions would bring with themselves a positive charge. Now, if I allow this to go unchecked, it will reach an equilibrium. We don't want to be at equilibrium, we want to be at homeostasis or steady state. And one of the differences is steady state requires the input of energy. And you're going to see why and how in a second. Now, there's another ion channel in the cell. And this other ion channel can allow a different ion to pass through the cell membrane, and that ion is potassium. And scientists measured the concentration of potassium ions inside and outside the cell, and they found that the concentration of potassium is much higher in the intracellular fluid than it is the extracellular fluid. Therefore, potassium will leak through this ion channel into the extracellular fluid. And of course, the rate of flow is based on concentrations and other things. Well, it turns out that Mother Nature in her grand design has another protein embedded in our cell membranes. This protein has, you know, as I've discussed before, sorry, let me pick this up. I've discussed this before, so slightly, and I want to reiterate this point. When we make a protein is a long chain of amino acids. And again, some of these amino acids have little side groups that like to bind to each other in chemical bonds. So if these two X's were to come next to each other, they might create a chemical bond, which might bring the two Y's close to each other, and then the protein folds up in this three-dimensional conformation. When a protein folds up, very often they have these little pockets that have amino acids that have charges that can bind to other substances. They can form chemical bonds. And so it turns out that one of the proteins that spans the cellular membrane has the ability to bind to both sodium ions and potassium ions. And it turns out that this protein has been shown to bind to 3 sodium ions from the intracellular fluid and 2 extracellular potassium ions. It's just the electrochemical properties of this protein. Now, when proteins bind to something, sometimes that causes 2 amino acids that were stuck together to release so that it can bind to something else. And what happens when it binds these sodiums and potassiums, it opens up a pocket to break down a molecule of ATP. And so I'm going to draw ATP here, and what happens is this protein will digest the ATP into ADP plus an inorganic phosphate. And as we discussed with ATP before, when I break that bond, there's energy that is released. And that energy allows this protein sort of to flip or pump the ions in the opposite direction that they were diffusing. And essentially what happens is it will kick those 3 sodium ions back out, and it will pump the potassium ions back in. And so the sodium-potassium ion exchange pump, or the sodium-potassium pump, is fighting this flow of ions. Sodium, due to its concentration gradients, will continually flow into the cell. And potassium, due to its concentration gradients, will continually exit the cell. Again, I don't want to let it go unchecked, or else I would reach equilibrium. And later on, when we study muscles in the nervous system, you'll see why that would not work for us. It would cause us to die. So the sodium-potassium pump is fighting this diffusion. As sodium diffuses in and potassium diffuses out, the pump keeps pumping them back where they came, so that these are always working. Even at rest, our cells are burning energy to maintain these imbalances of ions. So that's the sodium-potassium ion exchange pump. You should know that it pumps 3 intracellular sodiums back out of the cell and 2 extracellular potassiums back into the cell. And every time it does that, it breaks down a molecule of ATP. It's a form of active transport. Okay? Now, the other form of active transport that we're going to talk about is called vesicular transport. So let me erase all this mess. I hope you got it. You can backtrack and watch the video again and again and again until you understand it. But let me go on to vesicular transport and tie up this lecture so we can get on to the last little bit for the first exam. So now, as you guys should be aware, when we discuss the cell, and you watch the video on the organelles, within our cell and the nucleus, there are all these interconnected membranous tubules called the endoplasmic reticulum and the Golgi apparatus and other things. And it turns out that if I cut a section of this little membranous tubule out, that this membranous tubule can start to pull its membranes in. There are proteins that will start to pinch it across, and as it pulls all of this in, it can pop off a little bubble of membrane. And we call that a vesicle. A vesicle is nothing more than a little bubble of membrane that gets popped off of something. Or if something bumped into the cell out here, over time we would see the edge of the cell start to invaginate and take that in, and eventually it would seal off a little bubble of membrane, and it will have taken some substance into the cell. I could do the exact opposite. I can have a bubble of membrane pop off of here and have some substance in it. It will go fuse with the cell membrane, and as it joins the cell membrane, the substance is released out of the cell. These things can go in either direction. So when things bump into our cell, sometimes the cell can take them into a vesicle. And when things need to exit the cell, we have secretory vesicles that carry the substance to the cell membrane and release it. This is called vesicular transport, moving things around using these vesicles. Now, one of the things is it looks like it's kind of random movement around the cell, and that's not really true. If you recall, in one of our lectures, we talked about the cytoskeleton. So within our cell, there are these thin stick-like proteins called the cytoskeleton that give our cell shape, and they are very tiny proteins that form these long tubules that provide shape and support for the cell. But one of the things that we don't really get to see, and we're not going to discuss in great detail, but I just want to mention it to you, is that when I pop off one of these vesicles, all of these membranes actually have proteins in them. And some of the little proteins in these membranes can be bound by these other substances, these other proteins, and then those little proteins will move along the cytoskeleton so that we can deliver an organelle, a vesicle, to the cell membrane for secretion. Or if we take something in, sometimes those proteins, or those vesicles, are moved along our cytoskeleton. So our cytoskeleton not only provides shape for the cell, it is sort of a highway system for transporting things around the cell. Anyway, every time one of these little proteins is moving along our cytoskeleton carrying vesicles, it burns energy. They require ATP for this movement, and it requires ATP to take in a vesicle or release a vesicle from the cell. So vesicular transport is a form of active transport. And essentially, here's what happens. I talked about this before when we discussed lipids. Lipids like to stick to other lipids. And if you take a bowl of water and you put some lipids, some cooking oil in there, some vegetable oil, and you whisk it up with a fork or a whisk, it will break it into little small bubbles of lipids. If you wait long enough, the lipids that are trying to get away from the water will bump into each other and conglomerate into a much larger mass of oil. So we can pinch lipids together and seal off these little bubbles and have the bubbles come together and open up to each other and seal off still with their lipid coverings. So these vesicles can constantly be moved into and out of the cell and interact with other membranes. So here's what happens. If I have a cell membrane here, and I have a vesicle that has some little docking proteins that can interact with proteins in the cell membrane, and there's some substance in this vesicle can go and interact with some of these other proteins. And as it does so, over time, this vesicle, and I'm going to change the color of my vesicle membrane, okay, just so that we can see how it looks. It will start to open up by docking all these proteins together. And as it does that, whatever substance is inside the cell can be released. Anytime that we release a substance from the cell into the extracellular fluid using a vesicle, we call that process exocytosis. Exo means to exit or leave. Cyto means cell. And the osis usually means process of. So the name really tells you what it's doing. It's the process of something exiting the cell using a vesicle. So exocytosis is the active process of the cell releasing substances into the extracellular fluid using a vesicle. Again, it's an active process of the cell releasing substances into the extracellular fluid using a secretory vesicle. Okay? Now, I can actually do the reverse. I can simply reverse the arrows. When something bumps into the cell can actually invaginate and pull these pieces together. Again, this is all protein driven. It will pinch off a vesicle and pull it into the cell. And in that process, we call this endocytosis. Endo meaning into. Endocytosis is the process of the cell taking extracellular substances in using a vesicle, by forming a vesicle. So exocytosis is the process of the cell taking extracellular substances in using vesicles. Endocytosis has two separate forms. And these terms get thrown around and you should know them. One is called phagocytosis. Now, I never really liked this definition, but, it gets used. In phagocytosis, phage means to eat. So phagocytosis is the process of the cell eating. And one way that we define it is, this is the process of the cell taking in, and I put in quotes, solids, like other cells, bacteria, viruses, and other things, okay? So if a virus bumps into our cell, one of our immune cells, some of our immune cells can take the virus in and then fuse this vesicle with the lysosome and then destroy the virus or the bacterium or whatever thing gets inside your cell. Since we're taking in some kind of solid stuff, some kind of extracellular substance that has sort of a solidity to it, we call that phagocytosis. Now, what if my cell just takes in a vesicle that doesn't contain any solids? It's just extracellular fluid, just taking in fluids. That process is something that we call pinocytosis. Or some people say pinocytosis. So pinocytosis is the process of the cell taking in extracellular fluid using a vesicle. It's like the cell drinking. And when I was in high school, I used to get phago and pinocytosis mixed up. And then I realized, well, Pino's a wino, likes to drink a lot. So Pino the wino is cell drinking. Phagocytosis is the cell munching or eating, okay? Both of those are types of endocytosis, and both endo and exocytosis using vesicles is an active form of transport, okay? So now, if you've been following along, we should be done with page 18. We finished the top half of page 19, and I'm going to draw it to a close there. Again, I'm sorry this got broken into two videos, but it is what it is. I'm doing the best I can with what I got. I'm a low-tech guy. I'm an analog man living in a digital world, and I'm trying to get information across to my students and anyone else out there who finds this helpful. So I hope you had as much fun as I did. I hope you enjoyed it. I hope you learned something. Now, do this stuff till you can't stand it, do it till you understand it, and then do it 3 or 4 more times till you can explain it to a teenager, all right? I'll see you guys on the flip side in the next video. Thanks for watching.